Hi. Hi, hi, hi. How are we? I did, and I actually tried to look on Instagram, but I couldn't find you. Oh, no, need to take that off on my YouTube channel. Hi, I am good, thank you. I'm good. I say I'm good. I'm glad now that my um daughter's birthday party is all over, so I'm a bit more relaxed than I was on that front. Yeah, there's someone else with that name, so I was like, I'm not sure that's the one you meant. Hi guys, welcome. I'm on my TikTok and my YouTube, so I'm just kind of moving between the two. So if you let me just add in the thing, what am I doing now? I want to invite in case people want to join me. That's the reason why. Okay, I will have a look. Hi. Welcome. So if you want to join me on my Zoom, then please do click away. Hi, Paul. Oh, Alex, that's amazing. That's really, really good. Well done. I'm very proud of you on that one. So happy for you. And that's your starting point. That's the good thing. It's a starting point. Now it's like, where do you go from there? What do people want to do today? I thought I'd jump on here tonight as we're getting closer to the exam. I thought it would be good to make sure that I can give you guys as much help as possible. So if there's any specific questions that you guys have, please do ask away. Otherwise, we can look at some papers. Can you explain tissue fluid? Yes, of course I can. Of course I can. Give me 1 second. So if you are on my TikTok account, if you want to jump over, I am actually displaying my screen on my YouTube channel. So if you want to join me there. But if we look then at the tissue fluid, now they asked a lot in last year's paper on tissue fluid. So I think if they're going to ask a question on tissue fluid this year, it will be very much linking it to some specific part of the topic area would be where I would make assumptions for what that's going to be based on. But when we're looking at tissue fluid, it's important that you understand some key terminology. So you should know oncotic pressure, you should know hydrostatic pressure. So it's important that you note what's the difference between each of them. Well, from a beaten A during these last few, absolutely. It's all about the questions that you're doing. It's all about how you access the mark schemes so that you can get those keywords and terminology to get that across in the paper, is what I would suggest is important. As much maths as possible. Iris, absolutely. Hi, Andreas. Nice to see you. Okay, so let me just do this quick one on tissue fluid. I'm not going to spend lots of time on it, but I will jump around. So obviously you should know that you have these plasma proteins, these proteins which are able to be dissolved in the fluid, the water. So we're looking at water and its solubility property and understanding that that has a oncotic effect. So if you have these plasma proteins, normally you're we talk about albumin a lot, but there are obviously other plasma proteins. But the key thing for us to know is there's more proteins that are transported in the bloodstream than you would find in surrounding tissue fluid. So because of that, you have an oncotic pressure. That oncotic pressure is normally about minus 3.3 kilopascals. But if they're going to give you information in a question, they want you to take that information rather than just you pulling that number off the top of your head. You're not meant to memorize these numbers. What they want to see is whether or not you can pull the information out of what's been given to you. That's an application skill. And then apply that to the situation. So we've got these proteins, they're having an oncotic effect because of this oncotic pressure. So water moves into the capillaries by osmosis. We know that osmosis is the net overall movement of water. So it's really important that you understand this oncotic pressure is going to push the water back into the capillaries. So when we look at our arterial end of our capillary, what we note is the hydrostatic pressure is greater than the oncotic pressure. And because the hydrostatic pressure is greater than the oncotic pressure, that therefore means that fluid is squeezed out of the capillary and that forms your tissue fluid surrounding the cells. When we get to the venular end, because we're further away from the beating of the heart, because the pressure is less than it was at the arterial end, it's lost water, that pressure has changed. That therefore means that the water is going to move back into the capillary. And the reason it moves back into the capillary is because my hydrostatic pressure is lower than my oncotic pressure. So it's really important that you get that wording right. Because of the artery or something else, because so hydrostatic pressure is to do with the buildup of pressure when you have, so if you think about an artery, you know your artery has your endothelial layer, you've then got your elastic fibers, your smooth muscle, and you have your collagen fibers. So you know that it's got layers to it. So we know that water is incompressible. We know that we've got this structure that's being supported by the collagen. So therefore, when we talk about the hydrostatic pressure, whether we're talking about it in our arteries, our capillaries, our veins, whether we're talking about it in plants, if we're talking about hydrostatic pressure, we're talking about the pressure that's building up because the fluid has nowhere else to go. And when that pressure gets greater than the pressure pushing back, that therefore means that the hydrostatic pressure, because there's going to be hydrostatic pressure in both directions, it's about the net hydrostatic pressure. So there's always going to be a force pushing back, but it's about which one is greater. And that's why it's important that you talk about the hydrostatic pressure being greater than the oncotic pressure. So I hope that helps you with the explanation of the hydrostatic pressure. It's that incompressibility of water and the fact that the water needs to be pushed somewhere else because of the fact that we have these cells. And then if you're looking at the arteries or the veins, you have those extra layers which are keeping it. So Paul, if they're doing AQA, then they need to link more to the lymphatic system, but with the OCR, they need to know that the lymphatic system is a drainage system, that basically any excess fluid which surrounds the tissues will be drained away so that it doesn't affect the osmotic pressure of the cells. We don't want the cells to swell up and burst, because if the cells swell up and burst, obviously they're not going to be able to function and there is no wall to prevent them from bursting. So we need that drainage away of excess fluid, and that gets put back into circulation up at the superior vena cava. It's up at the clavicle here, where it's put back in circulation and then keeps going around and around. So that's the key thing they need to know about the lymphatic system. Andres, you've asked about paper 3, 2019, question 2 AII. So let me just open that one up and see where we're at. Question is, have we already done any of that paper? Have I got it here? 2019 paper 3. Oh, yes, we have. Let's see. I know you are, Paul. That I like the fact you feed me questions, because then I then go on, but because you've asked the question, I'm going to respond to you. Oh, right, here we go. We want to look at this paper here. So hold on, let me just check. Paper 3, 2019, you want question 2. Where did we go? Question 2, AII. Okay. So asking about the positive feedback. So in this one, it's a really interesting question. So hold on. What I'm going to do is I'm going to take this one and I actually want to move it across. to this here. And then I can start drawing a bit more. So what I will do is switch sharing and go back here a second. Okay, so when we're looking at this question here, the key thing for us to note is they want to know about positive feedback. So what do you know about positive feedback? Whenever you get a question like this, they expect you to be able to use your definitions and apply it to the question that's in hand. So if we think about positive feedback, then what we need to do is we need to think about the fact that we are actually increasing the magnitude of whatever the stimulus is. So when we look at, for example, in this case, what have they told us? They have told us that the core body temperature falls below 35 degrees C. So if the core body temperature falls below 35 degrees C, then what's going to happen? There is a form of positive feedback. So first of all, anytime you see, explain how positive feedback is, the first thing I would say to you is give a definition. If you give a definition of what positive feedback is, that's going to help you to get at least one marking point. So we have an initial change, which we know is our stimulus. Our initial change being our stimulus is going to be increased further. So we are increasing the magnitude of that stimulus. So the stimulus is going to be increased further. So if the stimulus is going to be increased further, how can we explain that? Well, what we've got here is they're talking about when the temperature falls below 35 degrees C, positive feedback causes this decrease in the body temperature to continue. So the stimulus was to fall below 35 degrees C. And what we're going to do is we're going to increase that. So when it decreases, this is going to lead to a further decrease in body temperature. And that was all in the question. That bit is just about you explaining what positive feedback is. Now, the next thing is that's then going to ask you to carry the question on because it's in explain how positive feedback is going to accelerate the process of hypothermia. So what is hypothermia? So hypothermia is where you've got a drop in your temperature, and that drop in the temperature is going to have an effect on the different parts of the body and how they function. Well, straight away, what do we talk about when we go to temperature? If we were looking at temperature and the effects of temperature, we would normally start to think about enzymes, and all students jump whenever we talk about enzymes to the end point of denaturation. And you'd be surprised how many students in the exam will have written that the enzymes become denatured, even though they're talking about below 35 degrees C. So we know if we're talking about humans that our optimum is about 37 degrees C, about there approximately. So what are we talking about here? If we decrease the temperature, we are now looking at a point down here below the optimum. So if we're at a point which is below the optimum, what is that going to do? So it's about the fact that what is it to do when we're looking at our body temperature? What is it that normally increases the body temperature? So when and you're absolutely right, we talk about our physiological responses, your shivering, your vasoconstriction, your hairs trapping the air. Absolutely right. That is a way in which your body is going to try to warm itself up, right? But we're talking that the temperature is low. What do you need to have, for example, the shivering to occur? For shivering to occur, you're talking about muscles contracting. For muscles to contract, they need ATP. Where does that ATP come from? That comes from respiration. So because you need all these things, if they are not happening, that therefore means that you're not going to be able to shiver if you don't have enough of the metabolic processes occurring. Well, what do all our metabolic processes rely on? They rely on our enzymes. They rely on the fact that our enzymes are going to be functioning at a rate which is optimum for our cells. So if we have a low temperature, straight away we should be thinking we have got less kinetic energy. If we've got less kinetic energy, our enzyme activity is going to be reduced. And if the enzyme activity is going to be reduced, that's basically saying that our metabolism is going to be reduced. So if the metabolism is reduced, if there is less respiration occurring, there's going to be less heat generated to warm it back up. Therefore, the temperature is going to fall further. So what we're talking about is less heat generated, and therefore it's positive feedback because it's increasing the drop further. And that's why it's hypothermia. As Paul's rightly said, hypo for Greek means below or beneath, hyper, above. So what we're doing is we're accelerating hypothermia because when it drops below 35 degrees C, there's going to be less kinetic energy, less enzyme activity, less metabolism, less heat generated to warm us back up. And that's what that question was looking at. Did you know that you were going to have an increase of that stimulus further due to the fact that that kinetic energy is the problem? Low heat, low kinetic. That's how when we put our food into the fridge or into the freezer, what we're doing is we are decreasing the kinetic energy of the enzyme activity of the microorganisms. And that's why we can store our food in the freezer or in the fridge and make it last longer, because we're decreasing that. Has that helped? I hope it has. I'm not sure if there's been any questions here or on the channel. I'm jumping around here, just double checking. Yeah, and that's the interesting thing, Andreas, in the fact that I'm shivering in the fridge. Yeah. Oh, what they do with questions like this is a complete application question. So straight away, that would be one of my go-to. The minute I see temperature, in my head, I jump to kinetic energy. And I need you to remember the part of kinetic. See, this is where I'm like, gotta slow down and write it the right way. Low kinetic energy. When I get too fast, it then comes out, my spelling gets so worse. But yeah, what I want you to understand is so many students miss that low kinetic energy part of the enzyme activity. So they automatically jump to denaturation for some unknown reason. And it's like there was a question in last year's paper where they asked about the polymerase, which is for PCR, and automatically students assumed that it was to do with the denaturation. Well, it's a higher temperature, so therefore it doesn't denature. Yes, that would be one marking point, but actually the fact that it can get to a higher temperature means there's going to be more kinetic energy, more enzyme substrate complexes being actually formed because of successful collisions, therefore a higher rate of reaction, because we haven't surpassed that optimum temperature. So it's really important that you don't just jump to denaturation unless they've told you that the enzyme activity has dropped down, or they have said in the question it's gone past the optimum. That's another thing to take your time when looking at the question. So the decrease in kinetic energy, who's just asked here, with regards to enzymes, you've got all particles have got kinetic energy. It's about how much kinetic energy they are given. So if we increase the kinetic energy of the particles and the enzyme, they're able to collide at a faster rate, and therefore we would hope we get a more successful collision. Enzyme and substrate are able to collide and bind together to give us the enzyme substrate complex, and that means that the enzyme can then lower the activation energy and the rate of reaction happens at a faster rate. So it's about linking it together and remembering that, yeah, absolutely, you talk about kinetic energy. Is this AQA? It's more OCR, but it does link to AQA. Good AQA in live. Lewis, I'm not sure who's doing lives right now, but there is a 75% overlap if it helps you. Do you do predictions? I kind of throw in some predictions whilst I'm going. So, okay, let me jump to mass flow in plants. I find that really an interesting one because they can go in different directions with mass flow. They can talk about the movement of the substances through the apoplast pathway, or they could link it to translocation. I do have a feeling, and this is where a prediction will come through, I do have a feeling that the translocation is going to come in, but not in the way that a lot of you will have memorized the topic area. I feel like the translocation may be a practical based one. I may be wrong on that, but I feel like there's something along those lines. Yes, I can. Lip of Henley on the kidney nephron. Grace, I'm not going to jump on that one. Grace, I would advise if you've only just come across my YouTube channel, go to my kidney topic area, so excretion playlist, and there is a very short video on that specifically. I don't feel like that one's coming up in that much detail because it came up not last year, but the year before. And I feel, but again, make sure you do cover it. I feel like it's not quite going to be as good, right? Let me do question 9 on paper 2 on the 2021. And then I'll go to question 10 on paper 2, 2022. So what did you say? Question 9, paper 2, 2021. 2021, paper 2. 2021, paper 2. And you want, I think that's literally just opened it at question 9 for me there. How random is that? What was question 9 you said, right? Hold on. Paper 2, let me just double check that. Paper 2, 2021, 2021. How random is that? I have just literally opened the paper up and it opened bang on question 9. That's so bizarre. Okay, so let's have a look at this question then, and then using the force. I'm not sure, it's one of those things because I do these papers so often with all my private clients that sometimes it's a case of I've looked at it or I've just had it open at that point was the last one. Okay, so this one is an interesting one because it's asking you about which one, and again, it's important that you really look at what they have told you in the question. So it's not. Which one is not required in DNA sequencing? So they're asking the question, what do you know about DNA sequencing? And therefore, what do you know is going to happen? So before I move on, I'm going to first look at what do we know? Well, DNA sequencing is where we're going to need to use DNA nucleotides. We're going to be using terminator bases. We're going to make sure those terminator bases have fluorescent, I hate spelling that, dyes, I think I've spelled that wrong, with that one. Why does the answer not say Taq polymerase? Because in this case, they don't expect you to write that it's Taq polymerase. So Taq is to do with the fact that it comes from the thermophilic bacteria. So the Thermicus, hold on, I've got to think this one through. Thermicus. I should know this off the top of my head. Aquaticus, I want to say. Paul, help me out on this one. Is it Thermicus aquaticus? Is that, have I got that one completely wrong on that front? See, this is where I'm like, I know it, but I know it's tack and I never, yeah, I know what it is. I'm wondering, because I used to know this. Is the name of the bacterium, which is what the tack, Thermus aquaticus. Oh, I was almost right. Thermicus aquaticus is the name of the bacteria. No, you don't need need to know that it's Taq polymerase. You need to know that it is a thermostable polymerase, but you don't need to know the name of the bacterium that it comes from, which is why I never memorized it or bothered worrying about it. So we do know that it's Taq DNA polymerase because we're going to do denaturation because we heat it up to 95 degrees C. We're then going to do annealing. Now, annealing is where we're going to use a primer, and that primer is going to be the starting point for extending. And in the extending part, this is where the Taq polymerase builds using the using phosphodiester bonds. Oh my gosh, I'm trying to write this so small it's not working. Hold on. Phosphodiester bonds. This is another thing that I would say to you as a student writing this. Now, obviously, I've been doing this for many years. I have been examining for many years, and half the time my students go, Christine, I cannot read your writing. If as an examiner, I cannot read your writing, even though I can magnify your answer. So I get a screen that looks like this, and I can magnify, but it doesn't necessarily mean that I'm going to be able to decipher what you've written. And if I really can't decipher what you've written, I will not be able to give you the mark. So one of the things I would say, if you're like me and you really struggle with slowing down your writing ability, not slowing it down. If you're really struggling, he's playing computer games right now and he's breathing, which is making me go, what is going on? Am I being too loud? He's got his headphones on, so he can't actually hear me. Anyway, so yeah, so where was I going with that? See, distraction, ADHD, nightmare. Yeah, if your writing's atrocious when you're doing your exam and I'm having to mark it, I will not I love that. Get back to the point. Yeah, I will get back to the point. I will not be able to read it. And if I can't read it, you're not getting the mark. So if you are really bad at writing or you know your writing is messy, slow down. Slow down because that's going to make a big difference for you. Okay, let's go back to this. We've had the extending. So we know we need our terminator bases. So we know it's not D. We know we need a primer. We know we need our DNA polymerase, which is Taq polymerase. So therefore, that would that would mean that the wrong answer for this one is C. So I hope that has answered your question. If you are a bio teacher and you're struggling and you're doing OCRA, my YouTube channel is built not only for students, but also for teachers, because I used to be head of biology and I knew how difficult it was to learn the content. So hopefully my videos are on playlist specific for your specification. I haven't seen the AS paper. No, I don't get to see the AS paper. None of my students sit AS, so I don't get to see it. It's so hard when time goes really fast. It is hard, but if you are someone who struggles and they haven't found a way in which you are needing that extra time, then that means that you are not entitled to it. You need to find a way to be able to get across what you need to say and to get it across in a neat way is what I would say. I'm glad you redacted your message. As much as I would love to switch to GCSE biology, because I know you've got your exam tomorrow, what I would say is there are lots of other videos out there for you. The reason I do A level now rather than just the GCSE is because there's so many people out there that are helping you. Right, what was the question? 22. That reminds me, 2019, 22. Where are you? Question two, 2022, question two, sorry, paper two, paper two, 2022, and I want question 10. Okay. I love the fact that some students jump on here and think that they're going to throw me off by saying some mean things about just get on with it and stuff like that. But the one thing I would say to you is we don't have to do this. We don't have to do these free sessions at all. And you're only setting yourself up. Okay. Why is A wrong in this one? Okay, so it's asking about, let's just have a quick read. Glycogen, large polysaccharide. Which option describes the structure and function of glycogen? So first of all, as I always do, I'm going to write some little notes myself. So I know that glycogen is made up of alpha glucose. I know it's got 1 to 6 and 1 to 4. And I know that it is insoluble. And I know that it is a storage for energy. So I'm just writing little notes myself to remind myself of that key parts. So 1 to 6 glycosidic bonds are more accessible to enzymes than 1 to 4 glycosidic. Okay, so Amna, the reason that that one is wrong is because what you're thinking is that the enzymes would be available to the 1 to 6. Now, if I draw, I don't even know what I'm drawing right now. Let's try that again. So if I draw a glucose molecule and I draw another glucose molecule, very basic. So there's my 6, okay? And there's my 1 to 4. There's my glycosidic bond. And then if I draw another glucose here, and I've got to think now because I'm going in a different direction, and I draw a 1 to 4. So there's a 1, that's a 1, and that's a 6. Here is a 4 to a 1. So Lynn, I can see you with the cholinogeric synapses. I will come to this one in a minute after I've done this question here. So the fact that it says the 1 to 6 glycosidic bonds are more accessible to enzymes than 1 to 4 is them trying to get you to make the assumption that there's no extra bits up here and that there's no coiling up. Remember the fact that it's branched, it will also compact into a small area, and therefore that means that the 1 to 4 and the 1 to 6 won't be easily accessible over the other one. It just depends where is that end part of any of the chains. So the end part of a chain could be a 1 to 4, or the end part of a chain could be a 1 to 6. So the thing about the breaking of the bonds being easier and much faster is to do with the fact that you are looking at that position at the end being accessible. And if that position is accessible, that will then allow for the breakdown. So on that one, what it was actually looking, so you understand why the correct answer is D, yeah? Perfect. Oh, definitely. There is lots of panic going on. I mean, it's crazy how much panic students go through, but you get it. You have got lessons. So 4 weeks' time, their first exam starts. So you can understand. And when I say the first exam for biology, they've already started the other topic areas. And so that's why there's a lot of stress that's going on right now. Right, Elle, what is your question with regards to cholinogeric synapses? I hate saying that because I always say it wrong. If I jump back to this one, what is your actual questions that you have on that one? Oh, nope, didn't want to do that. Let's try that again. There you go. So Elle, do you actually have questions for me to ask? So if you ask your questions, I'll then know where to come from when it comes to the synapse, or do you just need the content of it? Because if it is just looking at the content, then obviously, oh, okay. I'll stop talking. Whilst you're doing that, I'll look up the next question that I'm not asked for. What did you say? Question 20 on the same paper, 28. Question's that. 16. Oh, okay. We got a question yet? Right, Elle, if you type away your question, I will answer it when it comes up. I'm going to switch screens again and go back and answer this question. And then I'll come back to you for what you're not sure about on that one. Okay, so BIII. So here you're being told, haha, this is where we bring in Alan's awesome way of doing this. So you're being told you have one 0.01 centimeter cubed sample, and you've got 52 bacterial cells. So if I have a plate, and in that plate, what they're telling you is that they would count 52 bacterial cells. And the 52 bacterial cells that they're counting, they have assumed that each one of those represents a colony. Okay, so that's the important thing to remember, that they are representing a colony. Is representing one bacterial cell. So they've counted the colonies as 52. They've then assumed that that is 52 bacterial cells that started that colony off. Now what they're looking for you to do is say, right, so if I have 52 in my 0.01, this is my volume, the number of cells that I had was 52. So now what I need to do is I need to make my way back to work out how many were in my original sample, because it's asking me to say, describe the calculation step the students would need to make to estimate the total number of bacteria in the small flask. So to be able to work this out, I need to know how many serodilutions there were. So therefore, what I would need to do is have my 52, which is my bacterial colony. Now I know I've got 0.01, and that normally comes from, let me just double check what they've told us above. Here we go. And this is where you need to double check the information, that I started with 50 centimeters cubed. They then took that 50 centimeters cubed, and they were then looking at doing a serial dilution, and they've given you information here. So they're expecting you to look back at the stem of the question. The stem of the question starts right back at the beginning sometimes. So you should always go back to the beginning for this one. So it's not, you're right, it's not a tenfold, because this time instead, right, what we've got each time is 0.1 and 9.9. So we put 0.01 sample and counted there was 52 colonies. We know that they put 0.1 in with 9.9 centimeters cubed. So we know we have 10 centimeters cubed when they were counting. So we can see that. And then what we need to understand, to estimate the total number of bacteria, the students use a light microscope, and that's 0.1. They spread 0.1 centimeter cubed of the final serial dilution on the agar plate. So they've given you lots of information here. So it's really important that you're going back and looking at each of them. So we know we've got 0.01. We know that there was 52. So we need to do 52 times 1,000 so that we can get it back 2 are 10 centimeters cubed. So that's the first marking point, is to get that back to 10 centimeters cubed. So we have to times it by 1,000 to get it back to 10 centimeters cubed. And then what we need to understand is, because we're doing 0.1 centimeters cubed plus 9.9 centimeters cubed, we're therefore doing a 100 serial dilution, but we don't know how many dilutions that we've got. So this N represents the number of dilutions. So we need to multiply by the number of dilutions. And remember that they told us all the way back up here that it was 50 centimeters cubed. So remember, we're getting it back to 10 centimeters cubed. So we then need to times it by 5 so that we know how many were in 50 centimeters cubed. Amna, does that answer that question? Yes. Yes, it is. And it's about how many, because they've told you that you're doing 0.1 centimeters cubed and 9.9 centimeters cubed of water. Good. Right. What was the other question in this paper? And then let me just look at Elle's question whilst you're. A serial dilution, Andreas, comes up in every single year, just how they answer before that. Influx sodium ions is because of depolarization. How can it reach depolarization? Okay, so Elle, to answer your question, then I'll jump to the next one for you, Amna. To what you have to remember is your receptor cells are specialized cells that are going to be transducers, where they convert the stimulus, whatever the stimulus is, into a action potential, into a electrical impulse that is sent down through the sensory neuron. So the important thing for you to know, if we were looking at the Pacinian corpuscle, for example, what you have is the receptor. The Pacinian corpuscle is a specialized cell which role is to be able to detect pressure. Now, when there's pressure put on the layers, and the layers that you see wrapped around the Pacinian corpuscle, that is basically layers with lots of sodium ions. And what happens is, when the pressure puts down, that changes the shape of the membrane, which causes the channels to open slightly, allowing an influx of sodium. So if sodium can get in, and enough sodium gets in that causes depolarization, and this is the part that students get confused at. In fact, let me quickly jump screens and jump over to here. So when we talk about the resting potential, so here's my resting potential. We know, because I'm going to tell you, it's minus 70 millivolts. When we have any movement away from the minus 70 millivolts, we are depolarizing the membrane. Depolarization and depolarizing the membrane is where a lot of students get confused. So what we know is, when we reach the threshold value, whatever that threshold value is in your graph, what will happen is, at this point here, voltage-gated sodium channels are going to open. When those voltage-gated sodium channels open, there's going to be a massive influx of sodium. So that's going to make it jump right the way up to that plus 40 or plus 30 millivolts, whatever they decide to give you in the exam. So at this point here in the graph, when we are moving away from the resting potential, we are depolarizing the membrane. Only when it reaches the threshold value will those voltage-gated sodium channels open, And the action potential is an all or nothing. I want to move over here. Hold on. So going up, coming back down like that. So we have depolarization. We have our repolarization. We have our hyperpolarization. So the action potential is all those three things: depolarization, repolarization, hyperpolarization. When we depolarize the membrane and don't reach the threshold value, we don't generate an action potential. So therefore, that will drop down back to the resting potential. So depolarization, repolarization, and hyperpolarization are the three parts to an action potential. Whereas when we're talking about us depolarizing the membrane, that's when, before an action potential has been generated, that's whether or not you're going to reach that stimulus is going to reach the threshold value. So an action potential will occur, and it should then make its way all the way down, like you're doing this Mexican wave all the way down the axon. Bang, 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 bang. So depolarization, repolarization, depolarization, all the way down. The minute you've got one doing it, every other one after will follow suit. So once that threshold value has been reached, every single channel is going to be affected all the way down to the axon terminal. If you don't reach the threshold value, you're not going to have all the channels be activated. So therefore, you don't have to get the whole membrane back to the resting potential. Does that help? Um, no, the minute you hit threshold value, it's an all or nothing. It's not like you get, what I say to my students is I do a bit of an action. So bringing it up, so my hand's coming up as a stimulus. If I reach the threshold value, then I'm going to go woo, 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 and back up again, okay? So I'm going depolarization, I'm going repolarization, hyperpolarization, and back. So that moving up to begin with, I can move my hands up and back down again, up and back down again. I'm not going up, down, back, and forward. I'm just up and down. So if I don't reach the threshold value, then I'm not going to do 1, 2, 3, and back again. So it is really important to note that those voltage gated sodium channels will only open at that point that threshold value has been reached. They'll only close when you get to the top part, that plus 40 millivolts. Sentence really confused me. The influx of sodium ions into the postsynaptic membrane causes depolarization. An action potential in the postsynaptic membrane is generated. Yeah, it's because of that. That's the key bit that gets confusing. That depolarization and the depolarizing. So what would have been a better wording is the influx of sodium ions into the postsynaptic membrane causes the membrane to be depolarized, or causes the depolarizing of the membrane. But that depolarization, for most of you, you've just memorized it. That's the woo and back down again. So the depolarization, that influx happens, and it's a positive feedback mechanism here. We have a stimulus that sodium ions are coming in. They are therefore going to be increasing the number of sodium ions in by opening the voltage gated sodium channels. No, so it's about the depolarization, and then the depolarization that happens as a continuation. Once you've reached that value, you have then got to a point where all of them are going to go through it. So after the, no, Amna, be careful, because the confusing, it's not about them trying to be confusing. It's about the fact that we try and say things in different ways, which is why when you try and summarize things down, it can become quite difficult. And the reason I'm saying to you to be careful is because when you say an action potential, we're talking about depolarization, repolarization, hyperpolarization. All of those three together is an action potential. Resting potential, action potential. So depol, all three, one, two, three, that whole thing, all or nothing, it will either do it or it won't do it at all. It will just stay as that resting potential. Right, Amna, you said question 20 what? Let me jump across. 20 AI, right. Let me jump back. Where are we going? We're going over here. What time are we at? We've got a bit more time. Oh, I went too far, didn't I? Oh, here we go. What is the best method of memorization? Don't memorize. Sounds awful saying that, but memorization isn't going to get you to where you need to be. What you need to be is able to apply the content. Well, I have to say, as someone with ADHD myself, I'm actually autism. I'm still like trying to figure that one out. I would say, figuring out your weakest points and working through the hard bits that you hate. It's not fun. It's going to take a lot of effort, a lot of energy. And absolutely, Amna's right. Teach it to a friend, teach it to someone else. And even asking them questions of the finite detail that you need. So for me, I find writing like keywords down and only focusing on those keywords because my brain can cope with the keywords and then I can then embellish and I can go on different tangents with all the other parts. But I would definitely say it's not about memorization. That's not going to help you. I used to think that was true. When I was your age at school, I believed that that was the case, and I spent hours and hours and hours. That's not it. You need, if you have gone through the content, you need to have that application of how to apply it to the situation. So where's the intricate parts? Where's the bits that it's relating to, but it's kind of hidden. It's like a code. In the questions, there's codes, there's hints, there's little bits for you to pull out. You just have to practice and look for them. No, only get the action potential if they are if they've mentioned threshold value, then that's what I would say. Right, where am I? What am I showing you? I've got to double check what we're actually at now. Yes, perfect. Right. Question 20 AII. So let's see. They've got to love a bit of sampling technique here. So sampling is a good question to check whether you've done your module 4 content. A lot of people don't. So what is it that they're actually asking you? And let me just double check what question it was. It was this one, the first one. Right, so you have got a 20 by 20 area that you are going to measure. So we've got a 20 meter by 20 meters. So I'm going to do this. I've got 20 meters by 20 meters is my grid. Some students want to survey the abundance and distribution of the plants in the small area of the grass outside the school. What did they do? So they lay two 20-meter tape measures at right angles, starting in the southeast corner of the grass. They use a random number generator to select their X and Y coordinates. While facing north, place the left-hand corner of the quadrat on the point where the coordinates meet. Identify the species present in the quadrat. Identify, count, record in the table, new coordinate, repeat. Right. The teacher said that this method would not allow students to measure the distribution of plant species. So why is it not measuring the distribution? If the mark scheme says transect, why would this be more appropriate to use? Because there's no change. Like for example, there's no lake. Okay, so it's an interesting one. If I did my sampling and I've got my 0, let's say, hold on, 0 to 20, 0 to 20 meters. This is my sampling, and I'm going to measure, and I just happen to get coordinates that are like this. Am I going to get, and that could be completely random what happens of where I'm going to see, am I going to see the distribution of the organisms through that field? So you believe that that would show you the distribution of them through the field? So that wouldn't show me the distribution of them. That would show me what's in this corner. Whereas if I were to take a transect that goes all the way through the field or all the way across, and I was to measure systematically every meter, I'm going to look at the distribution on that transect. Yeah, does that help? So that's the reason why they want you to say, in this case, a transect. So the transect, remember, is systematic because you go 0 to 20, you're looking from one place to another. Because if there were, for example, a path where the students always walked, that would clear that area. And therefore, you would see that distribution going across. It could even be things like, it sounds ridiculous, but in GCSE exams, they tend to talk about the fact that you would have a bin. What would a bin do to the biodiversity in like a play park? So if you've got a field and there's a random bin that's sat there and it stays there for a while, you're going to have no growth around the bin because the bin's going to shade the area. You're going to end up with a different species that can tolerate not having much sunlight being there compared to other species, for example. So when they say a playing field, a small area of grass outside the schoolyard, that's one of the things they're looking at. So if I give you an example of outside my front garden, our neighbours every year get a slip and slide and they put it out in the front garden. It's all grassy area. And the way that it works, there's a bit of a dip to our grassy area. So what happens is all the water accumulates in the middle. And so therefore the plant species that grow in that middle area later on, due to the fact that they've had a lot of watering, is very different to those on the outskirts. There's also a path that goes around. There is no growth very close of the nice flowering plants close to the path compared to further on. So it's all about how we're affecting the distribution of these organisms by what is there. And it can be something as simple as, oh, in Cambridge, we're all about cycling. We are having cycle paths put left, right, and center. It's a good thing, but then it's a bad thing because obviously to get the cycle path down, you have to dig up what was there, the grass verges. What are you doing? You're affecting the biodiversity. So what do? They then plant new plants to try to bring the biodiversity back up. So it's like linking those things together. I think the grade boundaries are probably going to be about the same as they were last year. If you're trying to learn biology really quickly towards the end, as we are, what I would suggest you do is to spend time on your keywords. Make sure you know the main topic areas and the keywords that you can actually put across into answer the questions. Oxen investigations. Yes, give me a second. Application questions. So please, Abira, I'm not sure if you're still here. Question 18 and 21 DI of paper 122. Let me just have a look. Paper 1, 2022. 2022. And you wanted question 18. Oh, I went too far. Where are you? 18. 18. Just question 18 in general, you said. Okay, so again, back to how would you do serial dilutions? Yeah, it's really interesting, and it's very much linked to their module 6 population sustainability topic area, and the fact that you have to understand how, although we need resources for the growing human population, what we have to be aware of is the effect that we have on the biodiversity within these ecosystems, and therefore we need to think about how something would affect it. So there's lots of planning going in before we are able to just get rid of organisms to therefore make a new road or to have a new runway or whatever it is. Okay, let's have a look at this one. What are we told? So we've got potatoes, we've got coke boards. I love this because a lot of students assume that they are going to get a serial dilution question, and that serial dilution question is very much going to be about bacterial colonies and growth. And a lot of the time it's not, it's about other topic areas. And so students get a bit confused because they've memorized, and this is why I said don't do the memorization. They've memorized how to do serial dilution when it comes to counting colonies. And then they get a question like this and it completely throws them. They're then like, oh, what do I need to do? I don't understand. How do I do this? So what's really interesting about this type of question is they are saying to you, you have got a volume of 30, a volume of 30 centimeters cubed. You are starting with 1 mole per decimeter cubed. So what you need to do is if I want 1 mole per decimeter cubed, I am therefore going to put in the first one 30 centimeters cubed of my 1 mole decimeter cubed sucrose solution, and therefore I need to add 0 when it comes to my distilled water. So now I need to look at, right, well, I want 0.8, so 0.8, and I've still got 30. So if I were to do on the calculator that I want 0.8 of 30, how much would that be? That would therefore give me an answer, which is, double check this one, 24. Always like to double check my calculations. So if I've got 24 centimeters cubed of my sucrose solution, I therefore am going to need 6 centimeters cubed of my distilled water. Carry on with that. And I want 0.6 again of 30 centimeters cubed. So all it wants you to do now is say, how much am I putting in? So if I'm doing 0.6, oh, that's completely, oh no, it is right. 18, and then I'm going to put in 12. I'm going to put that the wrong way around, I thought, but I didn't. I did it well. And then what we're going to do is we're going to switch that around because we're now going to 0.4. So I'm going to do 12 and 18. Then I'm going to put 6 and 24. And then I'm going to put 0 and 30. So that's how to answer that question. And then the next thing it tells us on our next thing is to do with our uncertainties. Now, this is an interesting one because you were told that this weighing boat empty was 10.55. You're told that the weighing boat, when it has the sucrose, is 44.78. So you need to be aware that there is a difference between those two numbers. So you should know that when you're looking at uncertainty, you need to do the uncertainty divided by the change in whatever it is that you have been given. So the measurement. So you've already been told the answer that this is 34.23 grams of sucrose. They've told you how they did it, and they've told you that it was 0.01 of an uncertainty. So you should know it's 2 times 0.01 over my number here, which was my calculated value, which is 34.23. So therefore, I do 0.02 divided by 34.23, and they want me to work out my percentage, so I need to times that by 100, and that will therefore give me my percentage, which is 0.06. on that one. So that is, you need to be able to calculate percentage uncertainty and know that it's 2 times whatever they've given you here on that question. Okay, so what else have we got? We've got a table of results here. What can I see? Length, initial, final change, percentage change, mean percentage change in length. Okay, what can we see? Our sucrose concentration is going from high to low here. We've got a loss here. We've got a gain here. So therefore, somewhere in between, there was no loss or gain. So right away, what I'm doing is I'm just helping myself look at the data to see what it is that is telling me before I move on to the next question. They said the student was told that one of the results was an anomalous result. Right. Let's see. What have we got? Minus 1.5, 1.5, minus 2. Minus 3 minus 3 minus 4.1 minus 3.910002. Ah, interesting. That's a minus. Where's there all pluses? 4, 4, 5, 5. So therefore, if I look at that, I would therefore say that that one is my anomalous result. So my anomalous result is minus 2.9%, 0.4 third result on that one. And then if we look at state one variable that should be controlled in experiments and give a reason for your choice. Right, what did they change? Oh, I went too fast there. This is my independent variable. So I changed the concentration of sucrose solution. So because I'm changing the concentration of sucrose solution, it means I need to keep everything else the same. So I don't want to change that. I have measured the length and then measured the length as it changed. And the reason I've done percentage change is because I couldn't keep the initial length being exactly the same. So therefore, what other variables should I write about? And this is where I would say to you, have your list. Have your list your go-to on what to put down. Age, type, or species of potato. Why would that matter, Amna? So this is an interesting one because this is where students will have memorized a mark scheme answer, and that will then come up again. They'll try and regurgitate it, and it won't be asked the same thing. What do you mean about water potential? Good, yes. So if you used a potato that was a different age, then that therefore could potentially have a different water potential, which would therefore affect the results that you've obtained. So that would be another factor that you would have to consider. So if you can control the age of the potato or the type of potato or species of potato, then yes, that would be enough for you to control. Now, interestingly, when we're talking about temperature, for example, we think about kinetic energy. Does that have an effect on osmosis? Well, yes, it will, won't it? More kinetic energy, because there's more temperature, means there'll be a faster rate of osmosis. If we don't control the temperature, we are therefore going to affect that. So it is important that when you're doing these types of questions, that you're looking at what's my list, which ones are relevant to the question at hand. So age, type, species, and then your reason. What is it that would give the reason for it? Okay, what was the other one you said? 21 DI, 21. What we're looking at, 21 glands, endocrine, right? D. Oh, I love a question like this. So this is a really interesting one. So before I actually look at the question that it's told me, I'm going to really go through and take the time to decipher what information they've already given me. And that's the key skill I would say to you. For those of you who are asking me what, and it is a common question that's asked, what can I do to get better at application skills? Take your time reading the question. So Hypothyroidism is treated by taking tablets. Great. Scientists are investigating the use of stem cells. Right, straight away, stem cells, I'm just going to make a little note to myself. They can divide by mitosis and can differentiate. It's the question I would therefore also say, which stem cells are they using? If it is embryonic, then we know that that is pluripotent. And if it is adult, then we know that they are multipotent. So again, all I'm doing is writing little notes to myself to try and trigger my brain to help me to answer whatever the question may ask. To treat it in which the mice were destroyed using radioactive iodine, the scientists obtained mice. Here we go. So we're being told embryonic stem cells. So because we know they're embryonic, that therefore means that we know that they're pluripotent, unless they are within the 1 to 16 cell stage. Technically, I would say 8 to 16, because between that point's when they change. Without a mixture or growth factor for 30 days before transplanting into mice. Right, okay, so was the thyroid gland destroyed? No. Was the ESCs transplanted into the mice? No. So what have we got? We've got a control for comparison. What happens if we don't destroy the thyroid gland and we don't transplant the embryonic stem cells into the mice? If we do destroy it, but we don't do the transplant, what happens just from destroying the thyroid gland? Okay, so straight away, group 1, I can see we have a lot of variability in our data. We can also see, as it's told us here, 2 standard deviations. So each cross represents a single mouse, and the mean and 2 standard deviations has been given to us here. So therefore, I can see that in the data, and I can see that there has, by destroying the, so all I'm looking at right now is 1 and 2, by destroying this, what we have got is a lower thyroxine concentration that's being produced. Okay, so that's the first thing, that has dropped down dramatically. Right, what did they do to 3? Yes, they were destroyed. Yes, incubated in culture, but without the growth factors. Yes, it was destroyed. Yes, it was incubated and with growth factors. Okay, so we can see if it was giving them, but there was no growth factors, there's no real difference between the two, group 2 and group 3, with regards to the, although the mean is slightly higher, one thing to note is if I take my point here across, I can quite clearly see that there is overlap of my error bars. So I can see there's overlap here in the data. If I do give growth factors, there is an increase, but not enough to get me to the point at which I'm at the same mean as that. So straight away, all I'm doing is I'm pulling information, I'm deciphering what I can see from the graph, right? What is, and it was DI. The SCs, oh, they're described, look, they already told me they were pluripotent. So straight away, I had primed myself into what they could potentially ask me. What is meant by pluripotent? Well, I know that all stem cells are able to go through mitosis and then differentiate. The question is, can they differentiate to produce a whole organism or any type of cell? So that's what they're looking for, that they can divide and differentiate. And it's important that what we're saying is they differentiate into any cell type. The other thing to note, they're pluripotent, not totipotent, because totipotent are able to make a whole organism. So not, whole organism. So the significance of overlapping error bars, when we look up here, is that group 2 and group 3 Did not. So yes, they incubated in the culture, but they were without growth factors, whereas there was no incubation, no transplant, and no growth factors. So therefore, if we were looking at this data, although the mean is higher, there is significant overlap that we would say that there would be no significant difference because of the overlap. However, we would want to do a statistical test to be sure that there is a difference. Guys, my phone has now gone dark on me, which tells me I've been talking forever and it is now time for my bed. So that I'm going to stop there. I will try and jump on here tomorrow if I can after I've done my group session. Thank you for all the questions. Thank you for staying with me, for those of you who have been with me. And keep going with your revision. There is still time. There is still time. I know it's stressful. I know you've got other subject areas as well, but there is time for you to be making small improvements, getting those bits you don't know, so that if you're asked a question on that, to be able to improve. So thanks again, guys, and good night.